Good morning, everyone. I'm very delighted to have the opportunity to share with you guys the advancing reproductive health through innovative genetic analysis solutions here at Thermo Fisher Scientific. My name is George He. As Tamara had introduced, I'm the Director for Global Market Development and Marketing for Reproductive Health. At Reproductive Health, we want to be your trusted partner through three pillars. First, as you see on the left, is by offering innovative genetic solutions that enables customers like you to make future generations healthier. Second, we want to advance your business growth with a comprehensive menu of reproductive health solution. And finally, by providing reproductive health customers like you with our world-class support to make sure that you are successful in serving your customers. Let's dive deeper into our comprehensive menu solution as we discussed just now. As you can see here, it's a circle of reproductive life cycles, and we have comprehensive solutions from expanded carrier screening or carrier screening at 11 o'clock to preimplantation genetic testings, both for aneuploidy and monogenic disorders at 1 o'clock, to prenatal testing at 4 o'clock, newborn screening at 6 o'clock, and finally at postnatal testing at 8 o'clock. We see this as a continuous circle because any part of the information that you have gathered through these life cycles can provide meaningful information to decide on future pregnancies. You can learn more at thermofisher.com/RH. Let's go straight into carrier screening. Carrier screening by default is testing for autosomal recessive disorders, and these are typically inherited disorders and rare by definition. What does a rare disease means? A rare disease defined by NIH is a condition that affects less than 200,000 people globally. But collectively, they actually have over 7,000 of these rare diseases. So it's actually not that rare collectively. And as a group, because of these numbers, there are actually 1 in 10 individuals in the US that are affected with an inherited disorders. And these rare diseases are now a burden because over 35% of the mortality happens before 1 year of age are caused by these rare diseases. And over 18% of the pediatric hospitalizations are also caused by these rare diseases. So how are these rare diseases detected previously? These rare diseases and variants are bucketed into 2 buckets. On the left, sequence variants, and on the right, structural variants. Sequence variants are what you can think of as single polymorphism detection or multi-nucleotide variations. And some of the disease examples are like beta thalassemia or sickle cell disease and cystic fibrosis. Typically, sequence variants are detected using Sanger sequencing, mainly used for confirmation. They use genotyping or next-generation sequencing. On the right, structural variations are what we typically call copy number variations or deletions, duplications. Some of the disease examples are Duchenne's and Becker muscular dystrophy and alpha thalassemia. How it's been detected up to this point are mostly through MLPA or real-time PCR. So what you can see here is that different types of variants actually are being detected using different types of technology, which is difficult, cumbersome, and time-consuming. So what do we do? What do we offer here at Thermo Fisher to solve those challenges? We actually have 2 major buckets here, as you can see, either a CarrierScan, which is a flexible, scalable, high-throughput carrier screening solution, or the ones on the right, which is CarrierSeq, rigorously designed for expanded carrier screening research. Both addresses 2 very critical pieces, which is consolidations of standalone assays to improve lab efficiency, and it has comprehensive coverage from the content source that you trust and their pan-ethnic content. The major differences are the ones on the left. CarrierScan, it's a microarray solution with a very robust software solution, whereas CarrierSeq on the right is an NGS solution. So you actually got options to find the right tools for your lab's purpose. So let's dive a little deeper into CarrierScan assay here. This is a 6,000 manually curated relevant variant design on the array that covers 600 inherited disorders and our pan-ethnic content. They are designed through ACOG and ACMG database, so it's very reliable. It's something that everybody trusts. It includes multi-allelic and biallelic SNP, indel, and CMV. It avoids neighboring variants and SNPs and minimizing interference from pseudogenes. It's now tested actually over 1,000 variants, verified true positive samples here. Let's dive a little deeper here. Besides those, what I mentioned just now, I want to drive your attention to the honeycombs here on SMN1, HBB del dupe, DMD del dupe, and HBA1 and 2 del dupe. These are all typically harder to detect genes, and we're successfully incorporated all of that into our CarrierScan assay. And more importantly, we also have sample ID tracking markers to make sure that there is no cross-contamination or sample mix-up, which is an advantage in a large operations that people are pushing through 96 samples at a time. So in conclusion, CarrierScan assay, it's a high throughput, cost-effective, pan-ethnic targeted panel combining SMV and CMV determination with easy data reporting. It has the most comprehensive CMV carrier screening solution for challenging diseases such as SMA, DMD, and thalassemias. It also has over 6,000 variants that you can tailor and select the content that you want to report out. And it has a great software to go with it. So to learn more, you can go to thermofisher.com/CarrierScan. Doron Bahar, the CEO of Agenify in Israel, is a customer of CarrierScan, and he couldn't be happier after using it. This is his statement. It's an astonishing achievement to have a single assay with the ability to screen for thalassemia, spinal muscular atrophy, Duchenne muscular dystrophy, cystic fibrosis, phenylketonuria, and a plethora of hundreds of additional genetic conditions. And this ability is basically a part of the promise and fulfillment of the genomic revolution to a better health and better future. Now I'm going to switch gear and talk about the sequencing part of our expanded carrier screening research solution called the CarrierSeq Kit. This is a complete end-to-end NGS solution for ECS research. It's starting from genomic DNA extracted from blood or saliva, and it has the ability to maximize couples detection with full coding sequences and CMV analysis. It consolidates tests for difficult to sequence gene, just like CarrierScan, for example, like SMN1 here for SMA. It has the flexibility of multiplexing either 4 or 16 samples per run. And the simplicity of adoption with customizable reporting software is incredible. So what you're seeing here is an Ion Torrent ECS, CarrierSeq ECS kit, the partial gene list here. What you're seeing in the middle column of the target ClinVar SNV indels with the SC, those are actually special case genes with dedicated algorithm support. And the CMV column has a yes plus, means that we actually put in dedicated care to carefully design the SNPs around it to get a better detection of these genes. And these are just partial, the list of the genes that we have. What it comes out to be, it's a two-pool AmpliSeq panels. We went through 4 design iterations and tested with over 450 true positive research sample plus 68 synthetic variants. It's a full coding region of 420 genes, including 25 base pair flanking intron exon boundaries, and that ends up with over 28,000 carriers, SNVs, and indels from ClinVar. And as I was just mentioning, out of the 420 genes, over 30 of them has focused CMVs with extra amplicon designs to be able to detect the difficult to detect genes that was typically been shown very hard to achieve proper sequencing detections in all the homebrew assays, such as alpha and beta thalassemia, DMD, Gaucher disease, and SMA. So carrier status, as I was just mentioning, genes like SMN1s have high degree of homologies and paralogs. GBA has pseudogenes, and HBA has pseudogenes within the locus. So what we did was that CarrierSeq uses a combination of sequence read, depth-based copy number analysis, and SNV genotyping, plus specialized algorithm to call variants in 3 classes of these special genes. For SMN1, we target a copy number, and SNV genotyping analysis are used to determine the copy number of exon 7 with special analysis calling functional copy number. For HBA, it's a small gene distinct only in exon 3. So we actually use special algorithm analysis to resolve copy number across this homologous region. And then for GBA and CYP21-2A, with sequence homology, we basically use primary analysis, consider gene copy number and possible gene conversion event as mutation detection mechanism. So what you're seeing here is effectively the data analysis variant for the carrier screening or the CarrierSeq approach. So on the left is the Torrent suite software that you use to plan, monitor, and track your run. And then Iron Reporter is where the bulk of the analysis happens, where the calling of SNV, indel, CNVs, and gene conversion happens behind the scene. And then on the right, it's the Carrier Reporter software that automates the variant classifications for carrier analysis and reporting for individuals and couples. And this is where most of the lab people will spend most of the time looking at the variants at the end of the day. So the benefit here is that CarrierSeq has a full coding coverage of 420 genes with analysis of SNV, indels, and CNVs, and the outcomes that improve the detections of couples who are at risk of passing down serious conditions. And the benefit is that at-risk couple can pursue alternative actions such as prenatal diagnosis or IVF. For the difficult to detect targets due to homology such as SM1 and HBA in a single NGS assay, we, tremendously proud that we have actually consolidated all these standalone tests for technically challenged genes and improved lab efficiencies in terms of time and expense for ECS, and with optimized assay kits and software, which we then streamline implementation and help ensure reliable and consistent results, which then now most of the labs can now pick up ECS. And you can learn more at thermofisher.com/CarrierSeq. I like to continue on with our discussion into pre-implantation genetic testing in the reproductive life cycle. Why do we need to perform pre-implantation genetic testing for aneuploidy? Now, the benefit from this graph should be considered self-evident, but let me just walk it through for you. On the top blue line, the prevalence of aneuploidy in human blastocysts obtained in vitro is between 30% to 85%. If you look at the graph, at the age of 30, you have an aneuploidy rate of 30%, but when you reach the age of 44 to 45, the aneuploidy rate actually goes up to 85 degrees. On the middle green line, you have the risk of spontaneous miscarriage that happens at 10% around the age of 30, all the way to 65% at the age of 44 to 45. And the risk of aneuploidy in human fetus in prenatal diagnosis is between 0.2% at age 30 to 3% at age 44 to 45. So as women ages, it's important to be able to detect aneuploidy embryos to make sure that you are prioritizing healthier embryos for transfer. So what do we provide? We provide Ion ReproSeq PGS kit, which is a 2 to 3 hour hand time product from cell to data for 16 to 96 samples. You can see here they're divided into 3 major blocks of the workflow. First, on the Ion single-seq library, This is where you extract the genomic DNA, pre-amplify, amplify, and purify the samples. And this is where the bulk of the hands-on time is, anywhere from 75 minutes for 16 samples to 150 minutes for 96 samples. When you get to the middle part of templating using Ion Chef, the hands-on time drops down to 15 minutes, and the pipetting and the chip loading all happens automatically within the Ion Chef machine. And then when you get onto the sequencing part of it, again, the hands-on time is also only 15 minutes, where you only load the reagent into the Ion Gene Studio S5 systems, and you transfer the chip from the Ion Chef to the Ion Gene Studio sequencing to start sequencing there. So the Ion ReproSeq PGS kit for the Gene Studio system is for routine detection of whole chromosome aneuploidy and chromosome copy number event. It's scalable for 16, 24, or 96 samples, which gives you lower cost per sample, and it helps you combat price erosion. It has simplified instrumentation that helps you makes it easy to use and the ability to process more sample, and therefore scale your lab into increase sample volume and help reduce demands on your laboratory staff. It has a 10 to 13-hour turnaround time and improved data visualization and detection for mosaicism, which then gives you the confidence in interpretation of the results, and therefore you can also return results in a time-bound manner, more importantly, improve prioritizations of the samples. You can learn more about this product at thermofisher.com/IonReproSeq. I would like to bring your attention to Dr. Colin Lee from Alpha Fertility in Malaysia, who is extremely happy after using the products. As you can see in the middle here, we've been using Ion Torrent NGS now for about 3 years. The outcome is clear. We don't have many ambiguous or questionable results. And the scalability of the system is good, and as our experience with the system increases, so does our confidence in the results we obtain. With the volume that we're expecting to run before we bought the system has been far exceeded, because now they're very confident and they can run it very easily. The next one is an even more incredible story that really I think is important to highlight. Dr. Luis Alcaraz from our Spanish install base gone one step beyond and provided further innovation to combine PGT-A and PGT-M, which is for monogenic disorder detection, using the single biopsy materials for both using the AmpliSeq technology and the ReproSeq technology here. So he was able to just take one biopsy, divide the material into two, one run for the aneuploidy detection, the other one run for the monogenic disorder detection. And you can find out more about the product at www.pgd-seq.com. With that, I'd like to then further bring you into the next phase of the life cycle, which is in the invasive prenatal and the product of conception testing phase. So why perform invasive prenatal or product of conception testing? Invasive prenatal testing, according to ACOG committee opinion from December 2016, is recommended for a mother with a fetus that has one or more structural abnormalities identified on an ultrasonographic examination and who is also undergoing invasive prenatal. This is also recommended as a follow-up test for any positive results on screening tests. Product of conception, on the other hand, as we see in PCRS March 2017 publication, is that majority of the spontaneous miscarriage are sporadic and caused by chromosome abnormalities. So couples with recurrent pregnancy loss often undergo cytogenetic evaluation of the product of conception to better understand etiology. And this information then is important to use as a prevention for future pregnancy complications or miscarriages. How is it being done then for prenatal testing, invasive prenatal testing, or product of conception? Typically, it was done as in the left-hand corner called karyotyping. Karyotyping is a technology that's been around for a while, but it's a lower diagnostic yield technology where only about 3% of the cases of diagnosis made, and has a minimum deletion detectable of 5 megabases or larger. So anything smaller becomes much harder to detect. Actually, what's more difficult to this technology is the next two major points. One is the slower time to results, because if you are collecting cells from amniotic fluid, there is not enough material to detect. You actually have to grow them in cell culture to get more samples, more cells, in order to get the results. And that cell culture length increases the sample to result time from 7 to 14 days. Now, the next part is the error-prone workflow analysis. This is actually quite a difficult technology to scale, and it needs specially trained individuals, and the interpretation is very subjective. Let's just take a look at the graphics at the left-hand corner box there at the lower left. You see 6 different chromosomes there laid it out, but it's actually the same chromosome displayed 6 times. They look completely different, and without a trained eye, you couldn't tell one from the other. So that's where the subjectivities and the specialty needs of a trained individual comes in, and it's not scalable because each person can only do so many at a time. So what do we provide in to solve these challenges then is the CytoScan cytogenetic suite. It's a microarray-based detection technology. It has a much higher detection rate of over 3 to 15% increases in discovery yield. It has a higher resolution that can detect smaller chromosome aberrations, such as microdeletions or duplication. SNPs in the technology allows for detections of loss of heterozygosity, LOH or AOH, triploidy, and maternal cell contamination. Because it doesn't require cell culture, enough DNA just to use on its own, you can then get samples to result in just 3 to 5 days, so it's much faster. And because the analysis is done automatically using a software now, you actually avoid the subjectivity and the difficult to scale and the trainability of the person. So we make it much easier to use. You can learn more at thermofisher.com/pre-postnatal. So what does the suite include? On the left here, you see that the suite actually includes the cytogenetic microarray called the CytoScan chip. You have the kits reagent. You have the systems, which is the Gene Chip Scanner 3000 series, and more importantly, the chromosome analysis suite software. It's a software that's designed by cytogeneticists for cytogeneticists. So we know exactly what's required for the cytogeneticists to do their work, and therefore the software fulfills all of that. So how do we cover that? Cytogenetics suite is actually a genome-wide even coverage of SNPs and CMV probe. It's a future-proof content because it's already genome-wide. It removes any repeated validation required as new content comes online. It's also an ACMG recommended first-line genetic test platform, easy to use workflow, and easy to use analysis software designed by cytogeneticists for cytogeneticists, as I mentioned just now. The other component of the cytogenetic suite is a different chip type called the CytoScan exon array. This is focusing on comprehensive whole exome coverage of CMV and SNP probes. Its proven ability to detect single exon deletion duplication missed by whole exome sequencing, which we have a lot of customers who have done that and shown that there are just cases where the technology aren't capable to go down to. It also has the same easy workflow as CytoScan cytogenetic suite. Therefore, by running both, you don't need to add another full-time equivalent to run that. And the software is further optimized to specifically highlight ClinVar tier gene lists to help for prioritization of the analysis. You can find more at thermofisher.com/cytoscan. Again, I want to highlight some of our customers' testimonial of how they love this CytoScan product. This is Dr. Charlotte Andersen from Odense University in Denmark. She works with product of conception materials, And at every miscarriage, the DNA is usually very degraded by the time they get the samples. However, because the system is so robust, they are still able to get results, and means a lot to the parents if they can get an answer of what causes the miscarriage. The other feature that she really likes about CytoScan is the product where the CHAS, the analysis software, have both a CMV track and a SNP track. Now, if a deletion in the copy number variation track was detected, it can independently confirm in the SNP track, and therefore she doesn't need to go on to another technology to use for verification. So that's a beautiful added benefit to the CytoScan product to the cytogenetic community. Now, as I mentioned before, CytoScan or chromosomal microarray is a recommended first line for prenatal testing, as we discussed just now at the ACOG December committee opinion here. But Dr. Webner in 2012 also mentioned that chromosomal microarray containing SNPs allows for even greater discovery yield, as it can detect abnormality such as uniparental disomy, UPD, which is very important, that you cannot see on other technologies. You can also see triploidy in CMA, maternal cell contamination, and mosaicism. And looking at over 16,000 meta-analysis samples, we show that CMA additionally detects a significant aberrations in 6.9 cases of ultrasound abnormality, and 2.3% of all other indications. So that's a huge benefit to add. I'm going to switch gear a little bit to go to the newborn now after the prenatal. In the newborn screening research world, we're driven by two major diseases. First one is severe combined immunodeficiency, or SCID. Now, SCID is a severe genetic disorder characterized by a lack of cellular and humoral immunity. This incidence is about 1 in 40,000 to 75,000 live births, and it varied by genetic basis. However, it's 100% fatal unless it's treated early. So there is no question about that. It's important to detect SCID because it's 100% fatal. Children with SCID should not receive live vaccine because actually that's what causes the immune trigger response. Over 90% of the survival of the early hematopoietic stem cells transplantation will be able to solve this problem. So the test here is based on measurement of the episomal DNA product resulting from immune cell development, or for the T cells, which is TRAC, T cell receptor excision circle, and CRAC, kappa deleting recombination excision circle. The other condition is actually spinal muscular atrophy. And we talked about this at the carrier screening stage just now. Now, SMA is a degeneration of motor neuron in spinal cord and brain tissue in progressive neuromuscular weakness. It's the second most common fatal autosomal recessive disorder. It has a much higher carrier frequency of 1 in 50 to 60, and the incidence rate of 1 in 6,000 to 10,000 births. Now, what we are trying to detect here is the deletions of both copies of the SMN1 gene, so the homozygous deletion here. So what is our motivation to develop this assay then? The motivation is that in 2018, SMA was actually added to the recommended uniform screening panel, or RUFS, in addition to SCID. We also know that real-time PCR is already established in many of the labs researching in SCID because our Quant Studio is the gold standard of qPCR systems in the scientific community. Because we have two different block types of 96 and 384, is actually a scalable throughput for the reaction protocol. So for a medium throughput lab or high throughput lab, we have two different block types to help get through the throughput quickly. And we've been in the industry leader in multiplexing of qPCR assays. Therefore, multiplexing assays to analyze more than one condition will minimize additional labor or material costs. So what do we end up with? We end up with a Techman-based SCID SMA Plus assay, where you can go from blood punch to results with less than 20 minutes of hands-on time and only 4 reagent dispensing steps. And those are the 4: to wash, to lyse, to the assay and to run. Wash, we directly punch the blood card into the wells, and we wash them directly in there. And then we just then add lysis buffer to it to lyse the cell and get the DNA out. The third dispensing is by adding the master mix of the Applied Biosystems Tegman SCID and SMA Plus assay into the well. And then we put it into our Quant Studio system, and using our fast cycling protocol, we're able to get the results out very, very quickly. This is an example of results that you get for the SM1 deletion here. What you're seeing here is that in the blue area, the sample with homozygous deletion, you see no amplification. The black lines are the RNSP, which is your internal control to tell you that your sample actually amplified and there is DNA in there. So it's a very clear yes-no answer in terms of SMN homozygous deletion there. This is a plot to clearly identify SCID samples. What you see on top is the normal samples. At the bottom are the SCID samples. And what you're seeing, again, you have the black RNSP as a control, and then your blue and green of the TREC and CREC assays falling off without amplification on the lower box. So it clearly tells you that the samples did not have TREC or CREC assays, DNA, but you have SMN, which is a great another internal control to confirm the same samples there. So in conclusion, the Tegman assay for the newborn screening has robust quality. It's optimized for high specificity and detections of both SMN and SCID. It's simple. There's no DNA extraction, resulting in less hands-on time and lower costs. And with minimal liquid dispensing, the assay can be easily automated. The speed is fast because we can process more samples in less than 20 minutes or so hands-on time. And finally, without additional instrumentation needs, and us being the world leaders in qPCR reagents and consumables, we can provide the greatest cost savings to run this newborn screening for the genetic testing of SCID and SMA assays. Finally, I want to bring you back to the last part that we see in the reproductive life cycle, which is in the postnatal screening. Why do we need to do postnatal screening? This is a data published in 2014 to 2016. There was a marked increase in children diagnosed with DD, ID, or ASD. DD is developmental delay, ID is intellectual disability, and ASD is autism spectrum disorder. And you can see that the graph on the right just increases year over year from 2014 to 2016. In the case of children with autism spectrum disorder, there are research studies shown that the earlier you detect and diagnose a kid, the sooner you can bring out the maximum capacity of this child versus those who are diagnosed after the age of 4. Because the later they are diagnosed, it becomes harder to bring them out to have their full potential here. So how is it being done today in the postnatal cytogenetic testing world? If you are a DD or ID suspicious, you go through CytoScan as the first tier test on top here. And then you go through next generation sequencing. If there's only one hit, you add CytoScan exon suite to it to uncover the second hit. It's a long, drawn-out journey. What we're trying to propose is that instead of going through NGS and exon after CytoScan suite, you go and combine NGS and exon suite together. And we have examples from Children's Hospital of Los Angeles where they actually speed up the detection and finding much less questionable genes to go investigate much quicker. Now, on the bottom, if you have other suspected genetic disorders, instead of going through the same process of going through NGS or Sanger sequencing, finding one hit, and then add CytoScan exon suite, we are encouraging you to incorporate NGS and CytoScan exon suite together to speed up the testing journey here. So what do we offer? We actually offer the same product as prenatal using the CytoScan cytogenetic suite and the CytoScan exon suite together for the postnatal testing. Again, let me just reiterate CytoScan cytogenetic suite. It's a genome-wide even coverage of SNPs and CMV probes. It has future-proof content and removing repeated validation as new content comes along. It's HCMG recommended first-line genetic test platform, easy to use, workflow, and analysis. And then with the exon, it complements really well CytoScan cytogenetic suites and next-generation sequencing because it's whole exome coverage of CMV and SNP probes, and it has the proven ability to detect single exon deletions and duplications missed by whole exome sequencing. It helps finding second hit, and it's the same easy workflow as CytoScan cytogenetic suite. This one, I point you specifically to thermofisher.com/cytoscan exon to learn more about it. So Dr. Kristina Talames from Karolinska Institute in Sweden had done this research a few years ago, and what she really, she actually does both CMA and whole exome sequencing using our ion Torrent technology as well, and she finds that both CMA and whole exome sequencing are valuable in establishing molecular diagnostics in ASD. And with microarray, they can actually get to results fast and uncovering what's clinically significant, and the speed is important because it helps them plan the next steps in their studies. So finally, I want to bring you to this case example of Waardenburg syndrome here. This is a case where the mother has Waardenburg 2 syndrome, and the father has Waardenburg syndrome and a family history of Waardenburg syndrome. They unfortunately have a stillborn fetus at 32 weeks old. So they really want to find out what causes the stillbirth. So they ran it on CytoScan HD platform here in the US. They look at they found a large deletions across a SOX10 gene. But then when they want to find the second reason behind it, they weren't able to find it. Right? SOX10 is known to be a Waardenburg causing gene. So they ran a Waardenburg syndrome panel. They found nothing. They ran a whole exome sequencing across PEX3, all the genes that are listed on the right-hand corner column. They couldn't find anything either. So they ended up running CytoScan exon, and what they found was that it's actually a nested homozygous deletion within the SOX10 genes. So the resolution of the exon platform was able to give such high resolution that it enables them to find this nested homozygous deletion that we're not able to find with whole exome sequencing or other panel runs that they've done. So it's a great testament to how the technology helps move the research forward and find answers to the family who are looking for an answer. So I just want to wrap it out with the CytoScan portfolio of some of the histories that we have. CytoScan cytogenetic suite has been around since 2011. We've sold in 36 countries around the world, over 300 customers, happy customers who are using it. More importantly, over 2,600 publications cited so far. And if you look at the bar graphs at the bottom, those are individual year count. They're not cumulative. So it's amazing to see that a product that is so mature continues to generate so many publications year over year and increasing there. And the milestone that we achieved is that in 2014, we have received FDA cleared for CytoScan exon DX assay, and 2015, we've sold over 500,000 units of CytoScan HD. And then in 2018, we have launched CytoScan exon suite. So that's a great achievement here. Finally, I want to wrap this up with what I said on the third pillar of us being a trusted partner is providing world-class support. Now, all of this great product is not sufficient if we don't have a right support structure in place. So I'm very happy to tell you that we have now a follow-the-sun support model where anytime you reach a tech support, either through phone or through email, your case will be carried through until it's fully resolved. We know that in various types of labs, especially in the IVF testing labs, time is as an essence. And it's, some of people really need a 13 or 14 or 24-hour turnaround. So having this type of follow the sun support model really supports the important work that our customers are doing on a day-to-day basis. So again, I want to wrap it up. Hopefully I have told you about how we want to be your trusted partners by offering innovative genetic solutions that enable you, our customers, to make future generations healthier, by advancing your business growth with menu expansion, as I showed just now, from carrier screening to pre-implantation genetic testings, to prenatal, to newborn screening, and to postnatal. And finally, by providing reproductive health customers like you with our world-class support, hopefully we will be able to earn your trust to be your trusted partners. Just to remind you again, our lovely reproductive health life cycles of the 5 applications areas we're actively involved in. With that, thank you very much, and we will be happy to take any questions you may have.